This is a recap about enzymes and how enzymes work. First few points. All enzymes are proteins. As we know, proteins are made up of a long chain of amino acids that look something like this. So you can see there's a cartoon image of an enzyme, and here are all the little amino acids that make up that enzyme. Enzymes act as biological catalysts, which means they speed up chemical reactions that occur within the body. Living things have thousands of chemical reactions going on inside them all the time, and that's why they need these biological catalysts to make useful chemical reactions take place more easily within the body. So we'll have a look specifically at how enzymes actually work using the 3D model and the animation. So we've seen this before in class, and we'll have another look at it now. So this is sometimes called the lock and key model, and it explains how an enzyme and an enzyme substrate join together to break down or build up that particular molecule. So let's have a look. So here we can see you've got the enzyme itself, and this part here is the enzyme's active site. So you can see molecules that don't have the right shape will not join to the active site. So enzymes have specific reactions. They're really picky, and they usually only catalyse one reaction. This is because for the enzyme to work, the substrate has got to fit inside of this special shape here. So every enzyme has a unique shape that fits onto the substance involved in the reaction. So let's have a look. Here's the correctly shaped reactant molecule, or the substrate. You can call it a reactant or a substrate, and it fits into the enzyme like a key would in a lock. And together, when they're joined, it's called an enzyme reactant complex or an enzyme substrate complex. So let's have a look at what happens next. So chemical reactions usually involve substances either being split apart or joined together. Now in this particular example, you can see that something's been split apart or broken down. So this might be an example of an enzyme used in digestion. You can see what was the substrate or the reactant that fitted into this enzyme's active site has now been broken down, and you can see the two products here. If we have a look what happens next, the enzyme now releases the products from the active site, and the enzyme is now free to accept another reactant or another substrate. You can see this enzyme is left unchanged at the end of the reaction.